morning, everyone, both here in Lexington and in Princeton. Want to make sure, Princeton, that you've got your microphone muted so that we don't get any background noise or feedback during the presentation today. So it's my pleasure today to introduce our speaker, Mitchell Richmond, who is giving his exit seminar. Mitchell is from Olive Hill, Kentucky, over in the eastern part of the state, and he got his BS in agriculture and ag education at Morehead State University. And Andy, I still haven't figured out how he found his way from there to Princeton, but somehow he managed to get to Princeton. He worked on a master's degree here at the University of Kentucky and completed that a few years ago, and began working with Andy Bailey and myself on a PhD degree. And so today we're here to see the results of that effort. So turn the floor over to Mitchell.
So I don't think the mic's muted down at Princeton yet. You may want to mute that.
Okay. All right.
Thank you. All right. Well, thank you, Bob, for the introduction. And it's kind of crazy to think about the transition that I've made since starting at Princeton almost 6 years ago next month. I guess it will be 6 years ago next month. It's hard to believe that I've been at UK for 6 years. Maybe should be a 10-year seminar, or at least a 6-year review, or whatever that is. But I'm grateful that Andy and Bob took a chance on me and basically formed me in to find the career path that I wanted to, to be in, and that's to work with farmers, more specifically tobacco growers. And with their advice, I've landed a job up in Canada doing something similar to what they do here. So I'm very thankful for that. So today I'm going to cover a lot about one of the field experiments that we had since 2015, and I'm going to cover a little bit of the findings from another field experiment, and I'm going to go into a little bit of the lab portion of my PhD, where I collaborated with Dr. Yuan's lab and Pintu and Sanjay to help with some RNA sequencing. So Kentucky produces about 70% of the nation's total of Burley tobacco, with an estimated 5-year average yield of around 2,200 kilograms per hectare. And this is predominantly used as a component in the manufacturing of blended cigarettes, which I'm finding a little more out as working in Canada with blended cigarettes versus non-blended cigarettes, where they use 100% flue cured in their blend, well, I guess in their cigarettes up there, which are not really blended. But studies have shown that it takes around 150 to 200 hours of labor to grow 1 acre of Burley tobacco. So one of the things that we've been trying to focus research effort on, of course, is to reduce input cost. And one of those areas that we thought that we could reduce the input costs would be around topping. So you might ask, what is topping? Topping is the removal of the terminal bud or the inflorescence of the plant. And that is done on every plant in the field, which could be around 7,000 plants with Burley. And then also when you're topping, you're going through and you're breaking out the axillary bud growth, which we refer to as suckers. And those start to form at the junction of the leaf and the stalk. So as you can imagine, this is labor intensive and costly. So I looked for a very long time, and I guess it kind of goes to show that even know how much, I guess it doesn't matter how long you search, sometimes you can't find everything. And courtesy of Dr. Pierce, he was able to find this tobacco handbook from 1982 here at UK with Atkinson, Smiley, and Wallace. And so I looked trying to find an actual number of how long it took to top and sucker an acre of tobacco. And the numbers that they're reporting is it takes around 7 hours to top 1 acre and around 25 to 32 hours to sucker an acre, depending on sucker pressure. I think that might be comparable, but probably a little high as far as what we would consider an average. And I think for the most part, we would say around 6 to 10 man hours or labor hours per acre, which if you times that by the hourly rate, can be pretty costly. So after you manually top, traditionally you spray with some sort of suckercide afterwards, immediately afterwards, or the next day. And that can be with a systemic, a local systemic, or a contact. The only true systemic suckercide that we have available is maleic hydrazide, and the local systemic and contact have to contact each leaf axil to work. And that would be, for the local systemic, we use butylene, and which is a dinitroaniline or DNA. And I'll refer to that as butylene or DNA throughout the presentation. And we also used, and we also incorporated fatty alcohol, which is used as a contact to try to keep without using the systemic at first. And then we considered that purchasing and modifying equipment was not needed, since chemically topped plants would be sprayed anyway. So if you're in the field and you're manually topping, you're going to spray afterwards to kind of control sucker growth afterwards. Whereas this, we're just basically not topping and we're spraying beforehand to save the labor associated with topping. So I know this is kind of a busy slide, and I have a few of these today, but I just wanted to show some of the overall objectives, and I'm not going to touch on all of these today, but basically we wanted to determine if we could chemically top tobacco, and how does this compare to manually topping and spraying, and in terms of yield and leaf quality and sucker control. And to do so, we had to evaluate several different registered suckercide products and what rates to apply, and that'll be what I spend most of today's presentation on. And then if you look at 4 and 5, we wanted to look at differences between maturity groups of Burley varieties, and by that I mean time to flower, not time to harvest, and determine what plant growth stage that we could apply these treatments. And then the last objective, number 6, is where we collaborated with Dr. Yuan's lab and looked at RNA sequencing and gene expression with chemically topped treatments. So to get started, I want to talk about how we found what suckercide products and what rates that we should apply to practice chemical topping. So we started this back in 2015, and we concluded in 2017 at it started in Murray, but we moved that to Princeton. And if you ever drove from Lexington to Princeton and then to Murray and worked at all at both of those places and drove back, you would understand why we moved it from Murray to Princeton. It's to help try to catch those windows and not miss any applications. So it was moved from Murray to Princeton in 2016. But we used later maturing varieties, and that's based on some earlier work that was done by Bob and Andy, that some of the early maturities were kind of hard to time these treatments. But we had 2 manually topped treatments. Let me see if I get this to work. Yeah, 2 manually topped treatments. One was sprayed and one was not. So number 2 was sprayed, and that's what I'll refer to as the grower standard. So that would be where we manually top and then spray. And then one would be topped and then not sprayed, which would kind of be like your maximum pressure or a worst case scenario. And then we had 6 chemically topped treatments. 3 and 4 are with the systemic alone, like hydrazide or MH, at a full rate or a reduced rate. And then we had those same rates paired with the local systemic, which I'll refer to as full mix or reduced mix, and then the butylene or the local systemic, DNA, and then the fatty alcohol, which is the contact. Kind of to help with this is kind of the layout that I'll have in the tables whenever I show the results. Excuse me. So remember that the top 2 treatments were actually manually topped, and then the rest were chemically topped. And then I should point out that manually topped treatments were at 10% bloom, whereas chemically topped treatments were about a week earlier at 10% button. And you may ask, what is button? And I don't know how clear it is on here, but you can see the red arrow is pointing to what we considered a button, and that's where the reproductive growth was just above the vegetative growth without having to physically separate to find it. So let's get into some results. And recently I kind of like showing the result first and then showing the data of how I got to that conclusion. So today I'm going to follow something similar to that. And first I wanted to say that total yield and sucker control in most chemically topped treatments were comparable to the grower standard. However, we did find that we need MH and butylene or the mixed treatments because we were finding that MH alone in some cases didn't provide season-long control, and then we saw a reduction in total yield. Excuse me. So this is the same treatments that I mentioned, starting with the 2 manually topped treatments and the rest were chemically topped. But I want to focus your attention to the local systemic alone and the fatty alcohol alone. We saw that there was a reduction in sucker control effectiveness whenever we used these 2 products alone, and therefore we discontinued these because it was noticeable in the sucker control and the yield that these 2 treatments were not going to work, even though some previous literature that looked at chemically topping burley tobacco found that fatty alcohol was able to effectively chemical top. However, in the way that we were putting out these applications, it was not working for us. So I want to focus mainly on these treatments. And these would be the treatments that had MH with it and comparing that to the grower standard, which is where we topped and sprayed. And you can see that in most cases, we have equivalent yields, at least with the full mix and reduced mix. But we do have some issues with the smaller squares of where MH alone had a reduction in sucker control. Specifically at Princeton, I don't know really why that there's a difference between Lexington and Princeton, but as far as making broad strokes, we think that you should stick with the full mix, hard to find this thing, the full mix or the reduced mix treatments. And that was for sucker control. So now if you follow me along with the total yield, you see that the local systemic and fatty alcohol had a reduction in yield. It's quite well known that a reduction in sucker control will lead to reductions in yield because they are correlated. But again, I want to focus on these treatments, and more specifically these treatments that looks exactly the same as the sucker control effectiveness, and this is total yield in kilograms per hectare. And you can see that we're seeing a corresponding decrease in yield whenever MH is used alone compared to tank mixed with the butylene. So there is an added effect of the butylene, and across all environments, there was no difference between the grower standard and these 2 treatments as far as chemically topped yields are concerned. So that was a plus. That was exciting news all the way through the project that we're able to find somewhat of equivalent yields. Excuse me. There's no difference in leaf quality grade index, and leaf quality grade index refers to the value of the crop. We had a federal grader come in that was trained from the, I guess more of the auction times where we had all of the grades of tobacco, and they came in, graded the tobacco, and that gives us some idea of quality. And for the most part, the only significant finding is that the check in 1 year was significantly higher than all of the others. Don't really know how to explain that, but it happened. But in terms of comparing the grower standard to the chemically topped treatments, there's no difference across the board. So essentially we're seeing similar sucker control, similar yield, and similar grade index. So all things equal, we would save the labor cost associated with topping the plant. And it seems that chemically topped treatments tend to have lower maleic hydrazide residues, at least numerically. And I'll walk you through a table. And this is looking at maleic hydrazide residues for 3 treatments. And we did that because we thought that the full mix and reduced mix would be more of the treatments that would be used. And running residues is not the most cheap procedure. So we did focus our effort on these 2 treatments. And of what is significant, the chemically topped treatments have lower residues, but I don't really want to focus so much on that that we're not seeing higher residues. But it would make sense that we would see somewhat smaller less residues, mainly because we're spraying these treatments a week earlier than what we would be on the grower standard. And so that gives more time for rainfall and dew to help get those residues down. And the maximum residue limit for maleic hydrazide right now is at 80 parts per million, and we're well under that when you average over all the leaf positions. So I mentioned that more time for rainfall. Rainfall and MH residues are pretty well go hand in hand, and you can see that in our extremely low residue year at Princeton in 2016, we had the most amount of rainfall from topping through harvest. And then if you look at our lowest amount of rainfall in centimeters, we had some of the higher residues. So that is at least following somewhat of a pattern. But I did just want to point out that the grower standard in all cases had the highest, I guess, numerical residues, except for in Lexington at 2017. And that's likely due to the decision not to respray a treatment after we sprayed, and then within 3 hours we had a rainfall event. And we were afraid that if we resprayed, we'd have a lot more residues, but we just decided to take note of it and go on. I guess that happens with field work. And we spray this over the top of tobacco, so you might expect that the upper portion of the plant would have more than the lower portion of the plant, and that's typically, I think, what they find with MH residues. So, looking at tobacco-specific nitrosamine content, or TSNAs, we think that we see a topping effect and an MH effect, at least in one case, or at least in one location. And we're thinking that we also see that nicotine is lower in chemically topped plants. So I want to give a brief introduction to TSNAs. Tobacco-specific nitrosamines have been a focus of much research effort for the tobacco industry for some time. And these are formed from precursors that are formed from alkaloids. And with the curing environment, they form TSNAs. And TSNAs are, some of which are known carcinogens. So of course, keeping these at a minimum would be preferred. And then there's some previous work that has associated topping with an increase in nicotine content. So you might think that if you are not topping manually, that you may see a decrease. So to look at some of our TSNA data, excuse me, I wanted to point out that, of course, we're not seeing TSNA research is highly variable, and I did my master's in TSNA research in air curing and dark air cured barns, and there's a lot of variability there, and I felt like I was beating my head up against the wall trying to make sense of what data we're having. But you know, here I think this is the most one of the more interesting parts of the PhD project as far as the field work is concerned, because if you look at the Lexington location, we're seeing what I think an overall effect of topping versus not topping, where we're seeing a reduction in so the only difference between these two plots and the above plots would be manually topped versus chemically topped. And so I don't think that necessarily chemically topping is reducing TSNAs more so as the fact that we're not topping. But however, if I go back, this is not really the case for all of our locations. But we do see in the first, or in 2016, we did see, so this would be an overall effect. If you look at the UTC and the grower standard compared to the bottom two, that would be a main effect of topping. And then the difference between the UTC and the grower standard would just be an effect of MH. So in 2016 at Lexington, kind of got really excited that our data was showing this, because I was like, oh wow, we're seeing topping and an MH effect, which is kind of cool, right? So it didn't happen at any other location though. So I guess that kind of happens. But I think that we are seeing somewhat of an effect of topping. And if you look at the numerically highest in all cases, in all environments, it is the topped and sprayed versus the not topped and sprayed. So you might ask, how did that relate to the nicotine content? And I just want to briefly touch on nicotine, where we're seeing significant differences between chemically topped and manually topped. And again, there's a paper or there's a book that shows it's so 1990 that where topping increases the nicotine, and it's likely due to a combination of increased root growth, leading to an increase in nicotine biosynthesis, and possibly through the wound signaling pathway as you break out the top. But nonetheless, some interesting results that I think that a future graduate student would definitely have a project where if they looked at chemical topping procedures and manually top to see what's going on with nicotine and with the TSNA. And what I'm not showing is that we're actually seeing an increase in the conversion from nicotine to nornicotine, which is a step in the process. We're seeing an increase in conversion with chemically topped, but I think that's just more of where there's less nicotine, there's more being converted to nornicotine. So I've mentioned collaboration a couple times. Dr. Mark over in Ag Econ guided me through some economic analyses through some class and through discussion with him about this project, and we came up with a cumulative distribution function, or CDF, that compared manually topping burley tobacco to chemically topping burley tobacco. And this model is a stochastic simulation that used a fun distribution called the Jerks distribution, GRKS, which stands for Gray, Richardson, Close, and Schumann. And this is uses kind of a triangle distribution where you set the minimum and the mean and the maximum value, and then you do a simulation based on that, based on the data you provide. And I have that listed down there. But the main thing to keep in mind is if all else the same, and you remove the cost associated with topping, that you should save money based on that principle. And with the simulation, if we assume that it took 5 hours to manually top, we would save, based on our assumptions, around $130 per hectare, and then around $280 for assuming 10 hours. And the more time the more time it took to manually top, the more money you would save by chemically topping. So to give a summary of one of the field experiments where we evaluated different products and different rates, we found that total yield and sucker control in most chemically topped treatments were comparable to manually topping and spraying. However, you may need to add butylene to the mix because there is an added effect in two of the environments where we did see a yield reduction with MH used alone. And there should be no differences in leaf quality either, which would be related to value. And we are seeing somewhat of a reduction, or at least numerically, in MH residues. And the main thing is we're not seeing an increase in MH residues. And then I think we're also seeing, again, some interesting TSNA results with nicotine and conversion from nicotine to nornicotine, and an overall topping effect of TSNA, which with federal regulations proposing to regulate nitrosamine nornicotine, one of the nitrosamines, to 1 part per million, everything that's in the field that can reduce those levels may be of benefit and of company interest. So with that, I want to kind of switch gears and talk a little bit about the other field experiment that we had, where we had the same sucker side product and rate applied at different times on different maturities. And I won't spend too much time, but we had the same manually topped treatments. One was sprayed, one was not. And then we had 3 different timings of chemically topped applications, one at 10% button, one at 50% button, and then one at 10% bloom. And to kind of simplify this, we had the same late maturing varieties that we used in the other experiment, And then we also had a Tennessee 90, which is medium maturity, and then the 5 different timings or the 3 different timings of chemical top, and then the 2 manually top. So all chemically topped application timings resulted in basically success in terms of sucker control. However, we found that the 10% button and the 50% button should be targeted, because if you spray at 10% bloom, you're going to have at least 10% of those plants going into the barn with reproductive structures. So it doesn't necessarily halt. It doesn't revert. So if you don't physically break it out, it will still be there. And I just want to show a picture to kind of illustrate that. And I hope you can see there's some tops in there, and there's around 10% of this plot that still has a bloom. Even though the chemical top and treatment worked, you can see that it's kind of ill or dead. But if you sprayed it this late and you didn't go back and break those out, you would have that go to the barn, and that probably most farmers probably would not like that. Medium and late maturity groups are suitable. I feel like the late maturity group or late maturing varieties would be more forgiving of application because we found that not only do later maturing varieties reach that stage of switching from vegetative to reproductive growth later, but it also goes through those stages less rapidly. So in the field, you may have an extra day or two to get these applications on without missing the window, and then having to revert and go to manual top. And then most of these application timings resulted in good sucker control and equivalent yields and index compared to the manually top. So I won't spend much time on these data, but I just want to point out that, in the medium maturity, we were 91 to 100% control in all treated plots. The grower standard was, pretty well 100% or nearing 100% across the board, and we were getting something similar in most of these application timings. So it didn't matter as far as what time you sprayed it, the MH and the local systemic were going to work. And that's also the case for the late maturing. We did have one drop below 90 in the 50% button, but all in all, we were seeing pretty good control no matter what time you spray it, in the medium and in the late maturing varieties. So how does that equate to total yield? Total yield was not different between any of these, even in the years where we had, or even in the environments where we had more variability. Oh, wrong button. We still had equivalent yields as far as within any environment compared to the grower standard. And again, keep in mind that if you spray at 10% bloom, you would have 10% blooms at least going to the barn. And something similar to the late maturing variety, there were no noticeable differences between the two. It seemed like the treatments were working no matter what time you sprayed it in the medium or late. The only thing that I might suggest is to not use an early maturing variety, because it may be easy to miss that window. And this is looking at the grade index for the medium and late, and I just wanted to throw this up here to show that there's really no difference in grade index across any of the environments. And from what I can tell, it basically relates to the differences in environments, curing environments, and the years as far as the up and down in the grade, because it seems like they're pretty closely related. It just depends on which year they're in. So we're at 65 to 68 grade index here, and we're at 49 to 51 here. So the year and environment might have more to do. At least in those environments, it's pretty well the same as far as not different from each other when comparing the chemically top to the manually top. So a quick summary, again, all chemically topped applications were successful. Target less than 50% button, or at least before 10% bloom. And medium and late maturing varieties are suitable. And we didn't see much difference in sucker control, yield, or quality grade index for any of our treatments compared to the grower standard. And then I didn't show data for this, but generally chemically topped treatments had a shorter, narrower tip leaf, at the tip stalk position. We went in at stripping, or when we removed the leaf from the stalk, and we measured 25 leaves from each plot. And all in all, the chemically topped treatments had a reduction in leaf size as far as length and width. And that would only be important if, say, tip grade tobacco was more valuable compared to not, and whereas traditional topping, you may not get the size requirement. But that would be the only thing there. So a summary of the field experiments. All in all, I think that chemically topped tobacco may be a suitable alternative to manually topping. And as long as you use a tank mixture of MH and butylene, targeting the 10% to 50% button. And again, that's when the button is, the reproductive growth is visible above vegetative growth. And you shouldn't see any difference in yield, quality, or sucker control. However, there is a little bit less rapid change from vegetative to reproductive growth in the late maturing varieties, so they may be better suited for this. And then there is a high potential to reduce the labor cost, as I showed you with the CDF. Using the jerks distribution, we showed that around 130 and 260 plus dollars per hectare could be saved on average based on the assumptions in our simulation. So even though that this may be one of the, I guess, highly interesting portions of my project is where I had the collaboration with Dr. Yuan's lab and Sanjay and Pintu, and well, the three of them basically held my hand through some of the lab components of this, and where we isolated samples, and then we isolated RNA, and then we ran RNA sequencing. It was a really interesting part of my project, but I'm only going to talk about some of a real broad overview of some of the findings. So a short introduction to a small portion of this is maleic hydrazide and how does it work. So maleic hydrazide was first found, I think, in the late 1800s, but it wasn't until the late 1940s, early 50s that it was found that it had an effect on plant growth. And there's been a lot of different debate in the literature since then of how it works and what it does. But the commonality is that it seems like everybody believes that, well, and it makes sense, that it inhibits cell division without affecting cell elongation. Right now, if you open the WSSA, or Wheat Science Society of America, it's listed as a uracil antimetabolite. But there's been it's actually really interesting, and in my literature review for my dissertation, I kind of went down the rabbit hole of what is MH and how does it work. But it's kind of went through the ringers as far as what people say that it does and what it doesn't do. But anyways, that's kind of a brief, some of the things that people have said that MH does, and that's what currently is believed. So with that in mind, we had a couple of treatments that we imposed to where we had a control and then we had a chemically topped plant. So that would be not topped and sprayed versus a not topped and not sprayed. Sorry if this gets confusing. And then we had a topped and not sprayed, and then a topped and sprayed with MH. So this would be kind of the grower standard, and then this would be the UTC that we had in the other experiments, and then this would be a chemically topped, and then this would be kind of another control. And then after we imposed these treatments, we went back 24 hours later and collected axillary and apical meristems. And then again, with a lot of help from the lab, especially Pintu and Sanjay. So I wanted to show this figure that shows MH treatment significantly alters gene expression in apical and axillary buds. And you can see on the left, there were 132 upregulated genes, and sorry if you can barely read this, but this would be the apical meristem, which corresponds to this side of the circle. And then this is the axillary buds, which corresponds to the other side. But there's 441 genes that were downregulated in the apical and 132 that were up, and then around 2,200 and 450 up and downregulated respectively in the axillary. So what's interesting to me is finding, I guess, when you make a comparison such as this, there are 87 common genes where the yellow represents contra-regulated, which means that one was up, one was down. And then there were 8 that was commonly upregulated and 18 that were commonly downregulated. So again, I'm giving kind of broad strokes, and I just wanted to point out a couple of things. After gene ontology enrichment analysis, looking at how MH affects different developmental and metabolic pathways, just to mention a couple in the apical meristem, as far as upregulated, timing of transitions from vegetative to reproductive growth phase, and then downregulated with DNA metabolism and like cytokinin metabolism, and meristem maintenance, which kind of makes sense to me as far as what we're seeing in the field. And it also makes sense that in the axillary buds, the cell cycle and cytokinin metabolism would be some of the genes that were up and down regulated. And then what was downregulated in both, as far as, again, broad strokes, cell division control, which kind of aligns with what I saw in the literature review with how MH works. But not to spend too much time, because I know I'm nearing my time, collectively it's suggested that MH is suppressing DNA repair and recombination machinery, which leads to an inaccurate DNA replication and cell cycle arrest. And then moreover, MH inhibits apical and axillary shoot development, possibly by affecting cytokinin metabolic process in tobacco. So it does have a profound influence on gene expression in apical and axillary buds, and it's mainly with genes related to phytohormones, meristem development, cell division, DNA repair, and recombination after MH treatment. So I guess, what does all this mean in a roundabout nutshell is that I feel that chemical topping can be a tool in the toolbox for Burley growers. And I think that one caveat would be definitely uniform growth in the field, because once you spray your solution, there's no going back to retop, versus if you're manually topping, you can go through once, break the tops out, and then go spray a contact and then come back and spray again, so everything catches up. You don't get that option with chemically topped fields. And then again, target 10 to 50% button using a tank mixture of MH and dinitroaniline or local systemic. And then from what we've seen and under our conditions and our environment, there should be no reduction in yield or quality if you do these things as far as chemically topping. So with that, I would like to thank the village that it took to get me to where I'm at now. We were at a conference last year with Andy in Brazil, and it was hard for me to believe that over the last 6 years, the transition from cutoff shirts and I guess all of the attributes from being from Eastern Kentucky that kind of was worked out of me or worked into me, however you want to look at it. The transition that I've made, thankfully to everybody here, and especially these two men sitting up in the front, that have helped me along the way, and my committee, who has Dr. Aaron Haramoto, Dr. Mark, and Dr. Yuan, that at one point or another has found me at their office saying, "Hey, I'm lost. What do I do?" And everybody else, my wife Jenna, that has basically put me in this position to where I would have never guessed 6 years ago that I would have been here today. And I thank you for your attention, and the Jeffrey Fellowship for Tobacco Research, Altria Client Services, PMI, and JTI for funding this research. And with that, I'll try to answer any questions. Thank you for your attention.
Grade the leaves after curing compared to chemical topping. Grade them and you say quality.
Yeah, so the question was, did we, Princeton, yeah, Princeton's still there. The question was, did we grade the leaves after curing? Yes, so every plot that we we stripped out the leaves from every plot, and we baled them up, and then we kept them after stripping. And then we had a company representative come in, and we also had a federal grader come in to grade those, and that's how we were able to compare the leaf quality aspects. I didn't show any of the company data because it's a little bit more hard to follow, but the federal grade index is basically converted. They give us a grade, and then there's a citation that correlates that to a number, and then we're able to make those comparisons. So yes, they were graded afterwards. Does that answer the question? Sorry that I bored you all with a lot of data. Princeton, do you have any questions?
No. No more muting now.
Does that mean I'm off the hook?
Yes.